So let's compare prokaryotic and eukaryotic cells and also have a look at the organelles that are in the eukaryote cells. There's 3 syllabus objectives. We need to be able to compare the different types of cells. We need to be able to identify and understand the function of these key organelles, and we need to be able to identify the organelles using electron micrographs. So first of all, prokaryotic cells. So let's talk about this term prokaryotic first. A karyote is the Greek word kernel or seed, so we're kind of thinking about the nucleus. Pro means before, so it's before the nucleus. So prokaryotic cells don't have a nucleus, and they don't have internal membrane-bound organelles. So they're really quite simple and small organisms. They are always unicellular. So we've got bacteria and we have archaebacteria. All the biochemical processes that occur within the organism occur within the cytoplasm of this one cell. The DNA in the form of a chromosome is in the cytoplasm, and all biochemical processes occur in the cytoplasm. And they reproduce very simply through binary fission, splitting in 2. Now, eukaryote, U means true, so we've got a true nucleus. So eukaryotic cells are much larger and more complex compared with the prokaryotes. So we're talking about all of the other kingdoms. So we've got the protists, we've got the plants, animals, and the fungi are eukaryotic cells. They can be unicellular like the protists, or they can be multicellular like plants and animals and things. Now, what's really important here is that they have membrane-bound organelles. So all of these different structures here are specialized structures within the cell that have specific functions. And we're going to talk about those different functions in a minute. But we've got membrane-bound organelles, and that is where the biochemical processes occur. Now, they do have similarities though. Both prokaryotes and eukaryotes are bound by a plasma membrane. They both use ATP as their energy source. They both contain DNA in the form of chromosomes, and they have the same genetic code, which we're going to look at much later. But and they both have the ribosomes, which they use for producing proteins. So let's have a look at the organelles that are in eukaryotes. So first of all, there's not any particular order, but we've got the rough endoplasmic reticulum. This is what it looks like in terms of a diagram, and this is how we would identify it on an electron micrograph. We can see that we've got this highly folded membrane, and it's dotted with the ribosomes. So it's rough because it's got the ribosomes on it. And so it's a membranous structure that as a continuation of the nuclear membrane, and also is in contact with the cell membrane as well on the outside. Okay, so it's contacted with the nuclear membrane down here and the cell membrane on the outside. So it's a highly folded membrane that's involved in production, storage, and transport of specifically proteins. So we're going to contrast it with a smooth endoplasmic reticulum that doesn't have the ribosomes. So when we think about rough endoplasmic reticulum, we're thinking about it producing the proteins along with the ribosomes. So the ribosomes produce the proteins, and the rough endoplasmic reticulum stores and transports those proteins around the cell. So the smooth endoplasmic reticulum, it's smooth because it doesn't have the ribosomes. So instead of being involved in the production of proteins, instead it's involved in the production of lipids. Okay, so it's also a membrane-bound structure, and it connects with the nuclear membrane and with the cell membrane, but no ribosomes, and it's involved in the production of lipids. And so on the electron micrograph, you can see that it's also highly folded, but with no ribosomes. So just so you can compare them side by side. The Golgi apparatus, always spelt with a capital G, because Golgi is the name of the scientist. So again, it's a highly folded membrane. You can see it kind of wraps around a bit more, so it's I guess distinguishable from the ER, and it is involved in packaging and secreting. So you can see here we've got these vesicles that break off the Golgi apparatus, and then they move to the cell membrane and release their contents. So it's involved, this is like the post office. It packages and secretes products, cellular products. And so that's the Golgi. Next, we've got the lysosome. The lysosome is involved in digestion, breaking down of waste. So inside it, we have enzymes that are involved in, they're digestive or catabolic enzymes. So the lysosome takes in waste products and breaks them down. So they're quite small, so it may be hard for us to be able to see lysosomes, but they're certainly indicated here on this electron microgram. So mitochondrion is the singular and mitochondria is the plural. Mitochondria, of course, the powerhouse of the cell. It's a membrane-bound organelle, but it's also it's got an inner and outer membrane. So you can see here that's highly folded. You can see, you always can tell a mitochondria. Sometimes it's circular, sometimes it's this sausage shape, but you'll always see these internal membranes. So when we can see those, we know we're talking about the mitochondria. Now, these highly folded internal membranes are important because they increase the surface area for the aerobic respiration occurs across these membranes. Now, the nucleus often is the biggest organelle of a cell. You'll see that it's got a darker part in the middle called the nucleolus, which I'll explain a bit more in a minute. We've got this nuclear membrane. The DNA in the form of chromosomes occurs within the nucleus, of course. And we also have nuclear pores, which you may or may not be able to see, but it's the nuclear pores that allow ribosomes, for example, to move out of the nucleus. And so the other thing that will leave the nuclear pore is the mRNA, messenger RNA. So the nucleolus is a large subsection of the nucleus, and it's the site of the production of ribosomes, which are used for photosynthesis. So the ribosomes aren't really an organelle. They are the site of protein synthesis. And so we see that the prokaryotes also have ribosomes because they also need to produce proteins. So protein synthesis occurs at the ribosomes. Ribosomes are produced in the nucleolus and they move through to the cytoplasm. And in eukaryotes, they're bound to the rough ER and in the cytoplasm for the prokaryotes and also in the eukaryotes as well. So you can see we've got the rough ER here. We can see that it's studded with the ribosomes. We can see here that we've got the mitochondria. This is, of course, the nucleus, and that's a mitochondria here, and we've got the cell membrane. Now, the cell wall. So animal cells don't have a cell wall, but fungi and plants and bacteria do have a cell wall. It provides a rigid structure around the cell, where the cell membrane is involved in controlling what moves in and out of the cell. It's the cell wall that provides rigid structure. And so you can see the cell wall here, and you can see these plant cells are closely packed together. And then we can also, so just inside of that, we'll see the cell membrane, and you can see the nucleus here as well. Okay, so now we've got the chloroplast, which is the site of photosynthesis in plants. You can see this is a light microscope image, and you can see the chloroplasts here. And if we have a look at an electron microgram, we can see that inside the chloroplast, we can see these stacks called granum, and these are this is the actual site of photosynthesis that we're going to learn about when we do photosynthesis. So that's the chloroplasts and the vacuole. In plant cells, we have a really large vacuole, and that is involved in storage of water. Animals do have vacuoles, but you'll find that they're smaller, and they tend to be for the storage of waste in animals. So often quite easy to spot a vacuole on an image. The centriole you'll see with an electron microgram only. This is a tubular structure that is involved in cell division. So when we talk about mitosis and meiosis, we'll see that the spindles come out of the centrioles and they're involved in pulling the chromatids apart.